All right, if you're gearing up for the MCAT, you know cell biology can feel, well, huge, right? So in this explainer, we are going to cut right through all that noise. We're focusing only on the absolute highest yield concepts you have to know for test day. Let's dive in.
So, here's our game plan. First, we're going to lay down the foundation, the cell theory. After that, we'll take a tour of the eukaryotic cell. Think of it like a complex city. Then we'll see how that compares to the simpler prokaryotes. And finally, we'll get into viruses. You know, the ones that break all the rules.
Okay, so first things first. Before we can really get into the nitty-gritty of the cells themselves, we've got to understand the rules of the game, right? And that's exactly what cell theory is. It's pretty much the bedrock of, well, all of biology.
You know, this whole idea wasn't just figured out overnight. It was built up, piece by piece, over centuries. It all kicked off back in 1665 when Robert Hooke first peered at cork and saw these little boxes he called cells. Not long after, a guy named Van Leeuwenhoek actually saw the first living, moving cell. But the real game changer? That came almost 200 years later when Rudolf Virchow dropped a bombshell. He showed that all cells come from other cells. So, all that history boils down to these 4 core tenets. And for the MCAT, you got to treat these as absolute gospel. Lock these in. 1. All living things made of cells. 2. The cell is the basic unit of life. 3. Cells only come from other cells. And 4. They carry their genetic info as DNA, which gets passed down. It's worth noting, by the way, that this last one about DNA was added much later once we figured out molecular biology.
Okay, with the rules established, let's move on to the big players. And we're starting with eukaryotes. Now, I want you to get this image in your head. Don't think of a eukaryotic cell as just some blob of goo. Think of it as a sprawling, bustling city full of specialized neighborhoods and complex infrastructure.
So, you might be asking, why are we going to spend so much time on eukaryotes? Well, look at this number, 18%. That is a huge chunk of the biology questions you'll see on the MCAT. Seriously, getting this section down cold is non-negotiable if you're aiming for a top score.
So here's our map of the city. And the number one feature, the thing that makes it all work, is compartmentalization. See all these different structures, these organelles? Each one is wrapped in its own membrane. It's like having different factories and offices, each with its own walls, so they can all do their own very specific jobs, even jobs that might conflict, without getting in each other's way. It is just a marvel of biological engineering.
Right in the center of our cellular city, you've got City Hall, the nucleus. And if you're going to focus on one organelle for the MCAT, make it this one. It is so heavily tested. Why? Because it holds all the blueprints, the entire genetic library of the cell, in the form of DNA. And it's not just sitting there, it's protected by a double membrane, kind of like a fortress wall, with these little nuclear pores that act like super secure gates, controlling every single thing that goes in or out.
Okay, every city needs power, right? Meet the power plant, the mitochondria. Now, everybody knows it's the powerhouse of the cell, that it makes ATP. But the MCAT wants you to know how it does it. Take a look. It's got 2 membranes, but that inner one is all folded up into these things called cristae. This is a classic trick you'll see all over biology. Fold something up to dramatically increase its surface area. More surface area means more room for the machinery of the electron transport chain to work its magic.
But wait, there's more. Mitochondria are fascinating. They're what we call semi-autonomous. They've got their own little circular chromosome, and they can replicate all by themselves through binary fission. This is a huge, high-yield concept called extranuclear inheritance. And if that wasn't enough, they also hold the cell's self-destruct button. They can kickstart programmed cell death, a process known as apoptosis.
So our cell city is constantly making things and shipping them out. That's where the endomembrane system comes in. It's basically the cell's internal manufacturing and postal service. Imagine a protein that needs to be exported. The journey starts in the endoplasmic reticulum. Then it gets sent over to the Golgi apparatus for some final touches, and from there it's packaged up and shipped out. It's a beautifully efficient assembly line.
Let's look at the different stations on this production line. First, you have the rough ER. Why is it rough? Because it's covered in ribosomes, which are the little factories making proteins. Then there's the smooth ER. No ribosomes here. Its job is making lipids and detoxifying poisons. After that, everything goes to the Golgi apparatus, which is like the FedEx of the cell. It modifies, sorts, and packages everything into little bubbles called vesicles. And finally, the lysosomes. Think of them as the cell's waste disposal and recycling plant, full of enzymes to break down junk.
So,
a great question is, what stops this whole complex city from just collapsing into a blob? That would be the cytoskeleton. It's this incredible internal scaffolding made of protein fibers. You've got microfilaments, microtubules, and intermediate filaments, all working together to give the cell its shape and strength. But it's more than just scaffolding. It's also a highway system. Motor proteins literally walk along these tracks, carrying cargo from one part of the cell to another.
Okay, so individual cells don't just float around alone. They team up to form tissues. And for the MCAT, you really need to know the difference between two main types, epithelial and connective tissue. Epithelial tissue is the functional stuff, the part that does the main job of the organ. We call that the parenchyma. A perfect example is the nephrons in your kidney that do the filtering. Connective tissue, on the other hand, is the support crew. It provides the framework, the structure. We call that the stroma. Think of things like bone, cartilage, even blood.
All right, we've spent a lot of time in our complex eukaryotic city. Now let's pack our bags and head to a totally different world. A much simpler, more streamlined design, but wow, is it successful. I'm talking about prokaryotes, which, includes all bacteria.
This head-to-head comparison is MCAT gold. You're almost guaranteed to see questions on this. So what's the biggest difference? Prokaryotes have no membrane-bound organelles, no nucleus, no mitochondria, none of that. Their DNA is just a single circular chromosome, kind of hanging out in a region called the nucleoid. And here's a super high-yield detail. Check out the ribosome sizes. They're different. This isn't just trivia. It's the reason many antibiotics work. They can attack the bacterial ribosomes and leave ours completely alone.
When it comes to bacteria, they generally stick to one of three basic shapes. You've got the little spherical ones, those are called cocci. Then you've got rod-shaped ones, called bacilli. E. coli is a classic example. And finally, the spiral-shaped ones, which are called spirilli. Knowing the shape is often the very first step in figuring out what kind of bacteria you're looking at.
Now, one of the most important ways we classify bacteria is with something called the Gram stain. Here's how it works. Gram-positive bacteria have this really thick cell wall made of peptidoglycan. It's so thick that it traps the first purple dye we use, so they stand a deep purple. Gram-negative bacteria, on the other hand, have a much thinner wall plus an extra outer membrane. They can't hold on to that purple dye, so they end up getting stained with the second, pink-colored dye. And this isn't just for looks. That structural difference is absolutely critical for figuring out which antibiotic will work.
So, bacteria don't reproduce sexually like we do, but they are masters of sharing genetic information. And this is a huge deal because it's how they evolve so quickly and how things like antibiotic resistance can spread like wildfire. They have three main tricks. First is transformation, where they just slurp up bits of DNA from their surroundings. Second is conjugation, where they physically connect to another bacterium and pass DNA across a little bridge. And third is transduction, which is really cool. They get a DNA delivery from a virus.
Let's get a visual on this because it really helps. Over on the left, you can see conjugation. See that? It needs that direct physical connection. That little bridge is called a sex pilus. They have to actually touch. But on the right, we have transduction. There's no direct contact between the bacteria. Instead, you have that middleman, the Bacteriophage virus, that acts like a little genetic Uber, carrying DNA from one cell to the next. And that little viral delivery service brings us perfectly to our last topic of the day, viruses. These things are, well, they're the ultimate curveball in biology. They basically take the rules we just laid out with cell theory and throw them right out the window. Yeah, this quote really nails it. Remember rule number 1 of cell theory? All living things are made of cells? Well, viruses aren't cells. They're not even close. They're just a little bit of genetic code, DNA or sometimes RNA, stuffed inside a protein shell. That's it. No cytoplasm, no organelles, none of the machinery that we associate with being alive.
So if they're not really living, what on earth are they? The official term you have to know is obligate intracellular parasite. Let's break that down. Obligate means they have to, there's no other option. Intracellular means they have to be inside a cell. And parasite means they hijack that cell's resources for their own benefit. They're basically tiny pirates that can't do anything on their own. They have to invade a host cell and take over its machinery, especially its ribosomes, to force it to make more viruses.
And that's where we'll leave it, with this really big idea. Viruses exist in this weird, blurry space between complex chemistry and actual biology. They really make you stop and ask, what does it even mean to be alive? It makes you wonder, right? As we get better and better at looking at the microscopic world, what other bizarre things are we going to find that just completely shatter our definitions of life?